We are now going to begin the second half of Unit 2. So this is Part A for Unit 2, Part 2 PowerPoint. So we've already discussed within a cell, and we've discussed between cell generations. So this last half of Unit 2 is going to talk about between cells of the same generation. This is essentially horizontal gene transfer, where you have a donor cell, that means it's giving something up or some genetic material, and then contributing part of its genome to a recipient cell. There are three types of horizontal gene transfer that we're going to cover. The first one is known as transformation. The second one is known as conjugation, which I have mentioned before when we've talked about the pili. And the final one that we'll talk about is transduction. So let's begin. Let's begin by taking a look at transformation. So transformation, Griffith had an experiment that pretty much defined and explained transformation type of horizontal gene transfer. For this particular type of event, I would like you to think of the word dead or dead DNA from the environment. So in all three types of horizontal gene transfer, it's again, there's a donor cell giving up some of its genome, so some genetic material, and giving it to a recipient. So let's take a look at this experiment. You have pathogenic bacteria that's been injected into a rat. The rat has died as a result. This is color-coded as red. Then you have harmless bacteria. When something is harmless, that means it does not cause harm, and therefore this rat did not die. Then we had heat-killed pathogenic bacteria. So I want you to think of a time when there is a flood or some natural disaster. People are told to essentially boil the water to make it clean and get all of the pathogens and bacteria out of it. So here they took something that was pathogenic and essentially heat-killed it or sterilized it. Because it was heat-killed, when it was injected into the rat did not die. So in the last case, we have a mix. We have the harmless bacteria and the heat-killed one. So let's go back to the beginning of transformation and look at what grew on the actual Petri plates. For the pathogenic, it's color-coded red. It killed the mouse, and you see colonies of that particular pathogenic bacteria that's been grown by isolating from the dead mouse. Then you have the harmless one. The harmless strain is denoted by a purple color, and again, you have colonies of that purple harmless bacteria growing. When you have heat-killed bacteria, because you have killed it off, if you just compare the shapes of the regular pathogenic and the heat-killed, you could see that the shape in itself is changed. And then when you look at the Petri plate, nothing was actually isolated. There were no colonies because the bacteria was killed off with heat. Now when you take a look at the last one, where again you mix the harmless bacteria with the heat-killed one. So look at the pictures from before. Here's the red heat-killed pathogenic, and then the purple one, which is the harmless. Notice now on the plate you have both of them actually growing. Except take a look at what happened to the heat-killed. It essentially now looks like the pathogenic bacteria from the original plate. So when transformation occurs, what's happening is it is picking up dead DNA from the environment and incorporating it into healthy DNA within that particular organism and essentially bringing it back to life. So scientifically what's happening is genetic alteration is occurring through this transformation when a cell is picking up DNA directly from its environment and then expressing it. The dead part is the fact that it is picking it up from the environment. And remember, of the original injection, that also included the heat-killed pathogenic bacteria. So here's another picture of transformation to make it a little bit more visual. You have dead donor cell DNA. So again, this is the donor. Notice that it is broken apart. So now the DNA is in the surrounding environment. There is a live recipient that actually takes in. So if you look at the color here, that DNA from the dead one is red. So that DNA is now entering a live recipient cell. So here's the terms of donor and recipient. Once it incorporates it into its genome, from the environment, what happens is it starts disintegrating the donor strand and then incorporates it into the genome of the original strand. This is known as a transformed recombinant cell. So when you have transformation, you have a recombinant cell or a transformed cell. And again, this is picking up dead DNA from the environment and then incorporating it into a live recipient. The second type of horizontal gene transfer we're going to talk about is conjugation. In conjugation, again, you have this pili. Here it is right here. And this pili is almost like a tube. In lecture, I tend to talk about when you have an airplane in the military and a second one shows up that's a fueler jet. They pull that hose to mid-air fuel the other plane. So when this pili attaches the bacteria, what ends up happening is genetic material can pass between those two bacteria. One is the donor, which is giving up the DNA. The other one is the recipient. There are only two types of material that we're going to talk about, or genetic material that can pass. One is F plasmid, because it is the plasmid that goes through. If you think back to Unit 1, we discussed this. So the F plasmid is for fertility. And the second type we're going to talk about is the R plasmid. And we have almost an entire unit that is dealing with the R plasmid because R stands for resistance. So the first thing you should think of is antibiotic resistance. This is conjugation contributes highly to antibiotic resistance in bacteria. And I do want to point out, since you need to know this, of the three types of horizontal gene transfer that we talk about, conjugation by far is the least effective. And the reason it's the least effective is because this pili can become detached in the middle of the process of genetic material transferring. So it's hard to know what does or does not make it through, which is also why it causes resistance. So here's a visual representation. There's two types of conjugation, but essentially one is faster, one is slower, and they are a little bit similar. So here you have the bacterial chromosome, and here you have the little plasmid. This right here is your donor, and this right here is your recipient on the other side. What you should notice is that the two are attached, and they are attached by a pili or pilus. So what happens, if you take a look, this one is known as F positive. So if I asked you what type of plasmid this was, even if this wasn't written, you would know that it's an F plasmid, because remember, we are only going to deal with F and R. F is for fertility, R is for resistance. So this is F positive. That means it contains that F plasmid. This recipient is F minus, which means it doesn't contain that particular F plasmid. Through conjugation, this rolling circle mechanism, notice that the genetic material is passing from the donor to the recipient. And then if you look at the final picture, the donor remained an F positive, which means it did not give up its entire plasmid, just a portion of it, which then made the recipient also become F positive. So now both the donor as well as the recipient contain the F plasmid. The second concept of conjugation is known as HFR conjugation. And HFR is for high frequency. In this particular case, conjugation is occurring at 1,000 times the rate of regular conjugation. So, excuse me, this is the original picture from the previous page. Notice that plasmid becomes incorporated into this bacterial chromosome. So now this is essentially the genome. Then there's a pili. And again, through the pili, you have that rolling circle mechanism where some of this genome is transferring over to the recipient is sending it over, or the donor is sending it over to the recipient. And now both the donor as well as the recipient are going to have a portion of that genome passing over. And now you have this full recombination. And this again is HFR, high frequency recombination, making it at the rate of 1,000 times faster. In general, just a little piece of interesting information is it takes 100 minutes to transfer, so about an hour and a half, to transfer all of E. coli chromosomal DNA with the plasmid genes from the donor